guys, let's get going. Okay, ready? Let's do this. Come on. Okay, so we're going to talk about Dumas. So we talked about CalDolla on Friday. Those in lab saw both CalDolla and Dumas, and those in lab on Wednesday are going to see Dumas then. We're going to jump in on this. This method is also a crude nitrogen method, a crude protein method. And as we talked about on Friday, that means as measuring percent nitrogen also. And so how does it do this? Well, it does it through combustion of the sample. So we have complete combustion of the sample, and then it converts, and when you combust a sample, what different products do you get? What are the products of combustion? Carbon dioxide, water, and then what form is our nitrogen in when you combust a sample? Yeah, your nitrous oxide species, or your NOx species, you might hear it termed as. So what we're doing now is how this method detects percent nitrogen. It uses a thermal conductivity detector. And if you go back into your GC notes, you'll recall that what that does is you have a carrier gas, which we use helium, that's going to have a thermal conductivity, a baseline, that running over a filament. Then when you introduce a gas into that carrier gas, it's going to change the thermal conductivity of the detector. And that's what you quantify, is that change in conductivity, and you equate that to percent nitrogen. And so that means we need pure nitrogen in order to measure our percent nitrogen. So we can't have nitrous oxide, we can't have CO2, we can't have water in our carrier gas. So a lot of the method works to remove those components. So what happens first is we start here by combusting the sample. In the furnace, as you'll see, is you have, the furnace is at 950 degrees Celsius, and then there's an afterburner at 850 degrees Celsius to really ensure everything's combusted. So if you recall from CalDolla, that's about 500 degrees Celsius hotter than what CalDolla uses to digest the sample. From there, it goes through, and there's a filter here to remove particulates, and then this removes water from your initial gas stream. Then on this side, as those who saw in lab this morning, this is called the ballast. And so when the airstream comes in to the ballast works like a piston, so this moves up, and it mixes your airstream coming in. So it mixes those gases together, because just like we did in GC, we're only going to take a aliquot, or a dose, of that whole sample to be sent to the detector. And so the aliquot loops right here, and there again you'll see this better in lab. So it works on that similar principle of the GC idea. So it mixes in the ballast to get a homogeneous sample. From there then, it goes back up to here, and there's the furnace, there's a heater here, and in there this is a tube that has copper filings in it. And the copper filings do what then at this point in time? What? Sarah? That's exactly correct. So the copper filings is where we reduce now our nitrous oxide to nitrogen gas, right? Because we said we need nitrogen gas for our TCD detector. However, what's still remaining in our airstream at this point in time? Yeah, water and CO2. We haven't removed that yet. So that's where it goes up to here, to these scrubbers. And so the one is for your aliquot dose. The second one is for your helium here. Because, as we said, helium is our carrier gas to the detector. So we want to make sure that in both our sample moving through and in our helium, we don't have any CO2 or water vapor remaining. So they get scrubbed out there, and then they get sent to the detector. An advantage of this method is that we do get results per sample in approximately 4 minutes, which is great. A disadvantage is that you have to calibrate the instrument first. And if the instrument's running well, calibration might only take an hour and a half, and then you can have a very high throughput. As you see over here, we have what's called the carousel loading head. It's great. It finally works. It's been a few years since it's worked. We love it because you can put your samples in and walk away, and it'll just keep running them for you. Before, we had to actually manually push in the loading head every time we wanted to drop a sample. So it's great. We love our loading head. It's nicely automated, which is a major advantage of Dumas. But the problem is if you go back, there's a lot of things that can go wrong in maintaining it. Yep. As Cindy knows, Cindy has spent many hours maintaining this, and we're very grateful to have her do that for us. It's a lifesaver, literally. So there's a lot of things that can go wrong. One of the main common problems is, right, you have gas flowing through the system. So if you have any gas leaks at all, you're going to have an issue. And so trying to keep the gases moving through properly and keeping everything pressurized and the oven temperatures at the correct temperature are all challenges. So it's great, but it just has more maintenance than, for example, like a CalDolla method would have associated. This is another schematic of a Dumas that can be used. What I want to point out here, it's similar to what ours is, or the system we use, but you see here this mixing, you see the coil there? So what some Dumas methods instruments will actually do, instead of using, we had the ballast and then we had the scrubbers to remove our CO2 and our water. What they'll do is they'll actually use a GC column to separate your gases for detection. So basically, the idea there is then you're combusting your sample, and then you're attaching it to more or less a GC, and then that's what you're using to detect. Any questions so far? Okay. And like I said, you'll see this for group outs on Wednesday. You'll see this very visually in lab. So as with anything in the approximate analysis section, it's really important to be able to compare and contrast the methods. Because when you're out in industry or working or in academia and somebody says, oh, we have money for you. You always like those words. So they say, guess what? You can go ahead and you can buy some new piece of instrument to measure protein. Because they're getting into doing protein bars. And you need to measure your protein content now. How are you going to tell them which one to buy and why? So running through these. Principle we talked about. So this is something you just have to know the difference between. In simple form, CalDolla is using a stoichiometric chemical reactions to measure percent nitrogen, where Dumas is just by combustion and the separation of the gases. Instrumentation, which one is more automated, CalDolla or Dumas? Dumas. Which one costs more, do you think? Dumas. Yeah. What about time? Which one takes longer? That's some of those factors you have to play into it. If you don't have somebody, or if you don't have the ability to maintain the instrument and to pay those costs long over time, it could be more feasible for your group to use a CalDolla. Safety and chemicals. Which one's more dangerous? Yeah, you mean sulfuric acid, something you never want. And then when it comes to applications, as when we talked about just measuring protein, but there's also, actually Sonia had been looking into different nitrogen analyzers like the Dumas, and she told me that some of them out there can do what now, other than just measuring percent nitrogen?
They can measure sulfur and carbon. So it's like elemental analysis.
So that's one of those things where depending on what you're looking at, an instrument that can run Dumas like that might be beneficial for your lab versus just doing a CalDolla. So you can use it for different applications.
I was going to say, in terms of official methods of analysis, they're very comparable. Both are based on nitrogen.
Yep.
Determination, and both are official.
And they're both, to my understanding, I just verified this, the FDA says that you can use both for nutrition labels now. I don't think they used to allow you to use Dumas, but from what I've just recently read, the FDA will approve both for nutrition labeling. Okay. So moving on now, so moving away from our primary protein content measuring methods to a secondary method like IR, infrared spectroscopy. So what this does is based on the principle of measuring the vibrational energy of the peptide bond. As we talked about on Friday, that's a unique piece of the protein structure that we can then use to help us quantify. And if you go back to your spectroscopy lectures, you'll recall that IR is the wavelengths associated with the vibrational energy. What's nice is it's very rapid. You can use it in line. So this is the type of method you're going to do if you're trying to monitor the protein of a mixture running through the plant. You can have the IR in line, and you can keep watching your protein go through. Or if you're doing a membrane filtration and you want to quickly measure protein afterwards. When I was at Jack Links, they actually went through and they just got IRs for all of their labs. And they had to calibrate them though, because they wanted to make sure, because when you have meat coming in, right, your meat's going to have different fat contents. So if your fat content varies, your protein content's going to slightly vary. So how do you control to make sure that all of your jerky matches the nutrition label? So that was one of the things that they were doing, because they don't, in industry, most of them don't have a wet chemistry lab. They're not going to have a CalDolla unit in your plant. You're not going to afford a Dumas instrument. So you're going to send out to an outside lab to measure that for you. So if you want to monitor from batch to batch, you're going to aim to use a method like IR. And so the key with this one is, because of the peptide bond energy, we can use IR. So in addition to IR methods like this, so your secondary methods, we also have research methods. And so research methods are used for various reasons. Monday lab, what did you guys do in lab today that would be the research method? Bradford, exactly. Another research method that you can do is the BCA, or the bicarbonate acid method. So how this works is the proteins, because those protein bonds, as we talked about on Friday, can act as reducing agents. But in addition to the peptide bonds acting as reducing agent, if you remember from your structures of your amino acids from biochem, you have sulfhydryl groups, or SH groups, on your cysteine molecules, and on your tryptophan and tyrosine, you have OH, hydroxyl groups. And all those two functional groups can also act as reducing agents. So the combination of the peptide bonds and these functional groups then reduce, and you get a color change. And this color change is what you're measuring the absorbance of. So specifically for this method, the proteins are reducing cupric ions to cuprous ions. And that's what changes the reagent from, it starts out as a green color to this really pretty purple color. And the deeper the purple color, the more protein you have. As I'll talk about a little bit more after the Bradford, the thing with research methods is they're only really for comparison between samples. So for example, I use this method myself when I have all sorts of different samples that I'm maybe checking for a reaction that had happened in the sample. But if I'm checking for the reaction, so if you have a well plate, right, with all the little holes, or if you have lots of test tubes, and so if I'm checking for, let's say, maybe the Maillard reaction, or enzymatic browning in my sample, or lipid oxidation, but, well, not lipid oxidation, but let's say degree of hydrolysis, thank you. So if I want to know the degree of hydrolysis of my samples, well, in order to know how much degree of hydrolysis, I have to know the total amount of protein, right? And I'm not going to run, because of how the samples are prepared in solution, and you need purified solutions. Oops. You need a purified solution typically. For like example, the problem with this method is you have reducing sugars. They could reduce the BCA reagent instead of your sample, which is why you're going to centrifuge your samples first. But if I need to know the individual differences, I'm going to run the BCA. So if I have one well plate or one set of test tubes that tell me the degree of hydrolysis, I can correct using that same sample. So it's a way to check and balance in research, and it gives us a comparative idea between your samples, what's happening. So this is a BCA standard curve. This is mine, actually. I put it up because Amy and I have a competition going on who has the best R values. I did. I'm like, I'm going to put mine in there. Plus, my R value looked better than the one that was in there. Good friendly competition sometimes needed. So what we did for this one is we used BSA, which is bovine serum albumin, as our protein stock solution. And we then diluted the BSA from a range of 0 to 1,000 micrograms per milliliter. And what happens is then you add the reagent, and you're going to get different levels of absorbance. These are going to get different shades of purple. What then I can do with my samples, I'll add the reagent and I'll see where they fit along the line. So one of the reasons that these research methods aren't for like nutrition label quantification by any means, or their only comparison, is bovine serum albumin is a type of dairy, right? Yeah, it's a dairy protein. And it's not going to be the same as soy protein. So it's going to absorb differently than soy protein. Or it's going to absorb differently than if you have a meat protein. So if you have like a hamburger mixture that you're putting in and you want to see what's happening. So that's why it's only really comparative, because this is going to absorb very differently than your protein would, but it gives you a general idea. So this is one of the other research methods. This is the research method that you guys are going to do in lab. So as we mentioned, that some of the research methods work by proteins being reducing agents. The Bradford is a dye binding method. So what happens here, and this isn't in your slide, sorry. I added this one because of the lab piece, and the slides are posted. There's very little that was updated, but this is one of the things I updated, and the updated slides are posted. So how this works is the proteins are bound by a Kumasi blue dye, which then changes the color of your protein solution when the dye is added. Well, if the proteins bind the dye, it changes from a red to blue. So that shifts your maximum absorbance. So there again, you're measuring your absorbance of your solution. It's based on the amphoteric nature of the proteins. Anybody have any idea what that means? Any thoughts? What does amphoteric mean? Anybody know?
First, have you heard this term before?
Ampho means it's a bowl.
Anybody want to help them out? So it's this idea that an amphoteric, they can act as basically an acid or a base. Sometimes you'll hear it. So it can receive like protons or electrons, like to be able to exchange. And so the idea here is what happens is when you drop the pH, so you acidify the pH below the isoelectric point of the proteins, it then allows the dyes to bind electrostatically with the protein. And so that then changes the absorbance and the color. So all of these methods in the book does a better job. This is just an overview of comparing and contrasting when you would use the various methods. So for example, we have two research methods here, BCA versus Bradford. They both use a spec. They both just involve adding a reagent. How do you choose which one? Well, there's other things you look at that point in time. Do you have a method in-house for it? Do you have a method that you know how to use? Also, you have to look at your samples. So for the BCA one, it works by reducing. So if you have reducing sugars in your sample, it might not be the best method, where this might be a better method. However, for the Bradford, if your samples are too small, so if you have really small peptides, if you've hydrolyzed the protein, it's less compatible. It's not going to bind as well to those small peptides. So these are things you have to consider when you pick a method. So overall, you've seen this slide again. I won't go into it too much because we talked about this all already on Friday. But a key piece, looking at the research methods here, so you also see the Biuret and the Lowry method. They are other versions of the reducing method, like BCA. So all three of these work by reducing. And then Bradford is that dye binding method we just talked about. The book goes into more detail on all of these, so definitely read through that. The last method we're going to just touch base on is this UV absorption method. And so we talked about a little bit of this on Friday already. And if you recall, how this works is the fact that what type of amino acids are we looking at for the UV method? Ignis? Yeah, exactly. We're using the fact that we have aromatic amino acids to identify and then quantify, or I should say to detect and quantify our protein. So tyrosine and tryptophan, but also phenylalanine will, as the third aromatic amino acid, will absorb at an absorbance of 280 nanometers. Now, if we're in this UV range now, spectroscopically speaking, what type of interactions are happening in the UV? What? Yeah, electronic. So we're looking at the electronic changes, the absorbances, or electronic absorbance is what we're looking at here. So then here again, the concentration can be calculated then either using Beer's law, or you can generate a standard curve with your sample. What's nice here, it's non-destructive and rapid, but it has to be a very pure, clear solution in order for this method to work. If you have cloudiness at all or turbidity in your sample, it's not going to work. So what this is mainly kind of done for is if you have, let's say, samples coming off of an HPLC and you have a UV-Vis detector. This is a way that you can use that if you're detecting something else, but you want to make sure that your protein, you want to monitor protein too, this can give you a quick check. Or if you're looking for other things, right? There's other wavelengths we can be interested in for UV-Vis. It can look at like if our protein's conjugated or bound to a sugar. We can use UV-Vis to help us detect that. So if you want to make sure your protein's the same in all samples, it's really easy to just adjust the wavelength. So if you're measuring something at 340 nanometers, you can then quickly measure the same sample at 280 to see if your protein's comparative across the board. As a reminder, if you have small peptides, right? So if we hydrolyze our protein and we have small peptides, those small peptides might not have any aromatic amino acids in them. So instead, you might be looking at the peptide bond absorbance at 220 nanometers, which, as we mentioned on Friday, that can be less specific because when we're in the UV range, we're typically looking at electronic changes, which is easier to look at. It's more sensitive to look at your tryptophan and your tyrosine for electronic changes than it is to look at your peptide bonds. Any questions? Well, for those on lab on Wednesday, you're going to see more of this and specifically, and that's all I have. Pam, do you have anything else?